Good morning, friends. Myself, Richa. So, today I'm going to talk about the positive regulation of lac operon. Positive mechanism of lac operon. In this type of regulation is influenced by the presence of sugar, glucose. So, this is the very important question which is used to be asked, frequently asked in the CSR NET exam or NEET exam, in which they will ask whether when the glucose is low, what will happen, when the glucose is high, what will happen. So basically the presence of glucose here ultimately leads to the repression of the lac operon. You can see here when the glucose levels are high, there is a low rate of transcription. This you have to remember, that's it. So the vice versa happens here when the glucose level is low. Here you can see here when the glucose level is low, ultimately there is a high rate of transcription and translation. You can see here lac Z is encoding for beta galactosidase enzyme, lac Y is encoding for permease, and lac A is encoding for transacetylase enzyme here, which ultimately basically here you can see also leads to the production of glucose from lactose here. Lactose basically is divided into, it's a disaccharide sugar which converts into glucose and galactose. So basically it is also known as positive regulation of lac operon. Second, you can say catabolite repression. Catabolite repression. Is a positive control of the lac operon. So when the levels of cyclic AMP are high, you can see here the cyclic AMP levels are high, here purple color, readily binds with cap, that is blue color. So it will bind, which ultimately cap and cyclic AMP together binding leads to enhanced RNA pol binding, this, which ultimately leads to the production of these enzymes, beta galactosidase from lac Z, permease from lac Y, and transacetylase from lac A. So basically this binding is very important, cyclic AMP and cap, or you can say catabolite activator protein, which binds to a site near the promoter. Here it will bind, here, and stimulates the binding of RNA pol. So basically this cyclic AMP cap complex activates the transcription. You can make the star if you want to activate. Activation means star. So when cyclic AMP binds to cap, it will activate the transcription basically. This complex binds to the promoter here, right? So how this basically cyclic AMP is coming from? First of all, cyclic AMP is coming from ATP. ATP synthesizes cyclic AMP in the presence of enzyme adenyl cyclase. Okay. So the cap binding to DNA, when the cap cyclic AMP together will bind to near the promoter region, before before the promoter region here, right, causes the DNA to bend by more than 90 degrees. This binding enhances the transcription, by which more DNA will be available for RNA pol, which ultimately leads to frequent transcription here. Okay. So basically CRP, or you can say CAP, it is a cyclic AMP receptor protein, this blue color. It is necessary for high level of transcription. Alone it cannot able to bind, so it needs cyclic AMP. Together they can bind, they can form a complex which will bind to the DNA, and when the glucose levels are low, then there is a high level of transcription here, you can see. Now the vice versa, when the glucose levels are high, what will happen? Cyclic AMP levels are decreased in the presence of glucose catabolites. So when the cyclic AMP levels are very low, see here there are only 2 cyclic AMP here. Here there are more cyclic AMP. So when their cyclic AMP is low, so they will not able to bind to the cap here, so, which ultimately the bonding complex doesn't form. So RNA pol will not form here to the promoter. RNA polymerase does not bind to DNA as efficiently, which ultimately leads to the low level of transcription and the production of glucose is diminished. So when the glucose is high, here high means positive or something. When the glucose levels are high, then low rate of transcription will happen, and when the glucose is very low, then the rate of transcription is very high here. You can see here the rate of transcription and translation are very high. So basically, what are the elements of positive control here? Glucose, which is regulating adenyl cyclase, right, which is responsible for converting ATP into cyclic AMP. When the cyclic AMP is more, it will bind to cap. Cyclic AMP cap together will bind to the cap site, which is further facilitating binding of RNA polymerase at the promoter, which in turn causes initial transcription of lag Z, Y, and A genes. So this is the most important questions what they ask in CSR exam. Thank you so much.